Hi everyone, welcome back to another video. Today in this book reading session, all are welcome. Okay, so आज हम सेंट्रोजोम के बारे में पढ़ेंगे, ठीक है। सेंट्रोजोम एंड लास्ट में हमने यहाँ तक एंड किया था, ठीक है। 9/1/2026 में हमने यहाँ तक मतलब कवर किया। उसके बाद हमने पढ़ना बंद कर दिया था, बाकी दूसरे काम में एंगेज थे, बाकी दूसरे पढ़ाई भी होती है इसके साथ साथ। ओके, ठीक है। तो आज सेंट्रोजोम के बारे में हम पढ़ेंगे। नाउ मैं एक चीज़ कहना चाहता हूँ कि आप जब कोई बुक पढ़ो ना तो बेटर होगा, क्योंकि बुक में हम इस तरीके से एंड और ये टाइमिंग डेट ये लगा नहीं सकते हैं, क्योंकि बुक फिर गंदा हो जाएगा, ठीक है। तो आप इस तरह से अगर कोई डिजिटल डिवाइस, मेरे पास तो आईपैड है, अगर आपके पास पैसा है तो कीजिए, नहीं तो आप एक कागज़ में उसको भी लिख सकते हो कि यहाँ पर मैंने एंड किया है, ठीक है। इस तरीके से करने से क्या होगा, नेक्स्ट टाइम जब हम पढ़ेंगे तो हमें पता चलेगा हमारा प्रीवियस डेट में हमने क्या पढ़ा था, ठीक है। एंड इस चीज़ को बार बार रिवीजन करना, एंड हमेशा याद रखो कोई भी चीज़ में अगर आपको मास्टरी करना है ना तो किसी भी चीज़ को 10,000 बार करो। इट्स ए थंब रूल। कोई भी चीज़ को जब आप 10,000 बार पढ़ोगे, मान लीजिए ये जो चैप्टर है इसे 10,000 बार पढ़ना, तो तभी जा कर के इसमें परफेक्शन आएगा, ठीक है। एंड 10,000 बार ऐसा नहीं कि सिर्फ पढ़ते रहना, 10,000 बार अलग अलग चीज़ करना, ठीक है। 2 3 4 5 10 बार तो आप कॉमनली पढ़ोगे, उसके बाद उसकी राइटिंग करोगे, उसके बाद उसकी जो मीनिंग है लाइन बाय लाइन, सेंटेंस बाय सेंटेंस मीनिंग, फिर उसको प्रैक्टिस करोगे। तो ऐसे करने से वो चीज़ आपके दिमाग में, सबकॉन्शियस माइंड में रिटर्न हो जाएगा, ठीक है। सेंट्रोजोम। Centrosome is located closely to the nucleus in eukaryotic animal cell. It is formed by two centrioles placed at right angle to each other that are present within the amorphous pericentriolar material. Centrioles are short, cylindrical structure made up of microtubule placed in group of three triplet that run longitudinally in the wall of centriole. There are 9 set of triplets arranged at regular interval in the wall of each centriole. In the wall of each centriole. फिगर 4.10 कहाँ पर है? ओके, यहाँ पर है, ठीक है। Now the subunit of a microtubule in centrosome are gamma tubulin. Centrosome are microtubule organizing center, that is microtubule organizing center. दूसरा, अब हम देखेंगे सेंट्रोजोम का क्या कुछ स्ट्रक्चर है, ठीक है। तो सेंट्रोजोम रेगुलेट क्रोमोजोम मूवमेंट ड्यूरिंग सेल डिवीजन। सेंट्रोजोम रेगुलेट क्रोमोजोम मूवमेंट ड्यूरिंग सेल डिवीजन. They duplicate themselves and move apart from each other to the pole of a mitotic spindle to monitor the process of a cell division. Now, transverse section across a centriole. Note 9 group of tubules, each group having 3 microtubules. तीन, तीन छ, तीन नौ, ओके। साइटोस्केलेटन, साइटोस्केलेटन, ओके 1 मिनट। Cytoskeleton is an intracellular system of fiber that not only maintain the structural integrity of cell but also allows appropriate change in cell shape for cell mobility and participation of cell in various physiological activities. It consists of a microfilament, microtubule, intermediate filament. These cytoskeletal elements are made up of different cell protein. Table 4.4। ओके, आगे चल करके है माइक्रोफिलामेंट। All eukaryotic cells contain microfilament that contribute to maintenance of and change in cell shape and regulation of cell function. These are long solid filament, figure 4.11a, having diameter of about 7 nanometer. They are made up of actin. Actin is the most common cell protein that accounts for about 15% of total protein in the cell. Actin in the globular form is called G-actin, which is the unpolymerized actin subunit. G-actin subunit polymerized to form filamentous actin referred to as F-actin. In vivo, actin filaments polymerize and in vivo actin filament polymerize and depolymerize. Also, polymerization occurs at one end of actin filament and depolymerization occurs at other end. Special characteristics. Those microfilament are well developed in muscle cell. They are present in almost all cell and better organized in cells that secrete granular content by exocytosis. For example, the developed contractile system in platelet consists of microtubules and extensive network of filament. Microfilament help platelet to change shape and move granule from interior of cytoplasm to canaliculi for release of a chemical, that is release reaction of platelets in cell with microvilli on their epithelial surface. Microfilament extend into the microvilli. Function of a microfilament. Microfilament are the major contractile fiber of the cell that helps in the change in cell shape and cell movement. As microfilament exhibit contractile phenomena within the cytoplasm, they help in phagocytosis, transport, transport and secretion of a cellular material. 23, 24, okay 22. secretion of a cellular material, cell contraction, cell discharge, movement of a secretory granule etc. नाउ टेबल 4.4 हमने प्रीवियसली पढ़ा था कि 4.4 देखेंगे. सो प्रोटीन सब यूनिट एंड डायमीटर ऑफ अ साइटोस्केलेटल एलिमेंट ऑफ द सेल. पहले हम देखेंगे माइक्रोफिलामेंट, दूसरा हम देखेंगे इंटरमीडिएट फिलामेंट. सो पहले दोनों का शेप देखेंगे. माइक्रोफिलामेंट का शेप होता है डबल स्ट्रैंडेड हेलिकल अरेंजमेंट, जबकि इंटरमीडिएट फिलामेंट का शेप होता है ट्यूबुलर हॉलो. डायमीटर होता है माइक्रोफिलामेंट का 7 नैनोमीटर, जबकि इंटरमीडिएट फिलामेंट का 10 नैनोमीटर. बेसिक प्रोटीन यूनिट इसमें माइक्रोफिलामेंट में एक्टिन होता है, जबकि इंटरमीडिएट फिलामेंट में वेरियस प्रोटीन होता है. लोकेशन इन सेल की अगर हम बात करेंगे, माइक्रोफिलामेंट फॉर्म से नेटवर्क एडजेसेंट टू सेल, कोर ऑफ अ माइक्रोविलाई, कॉन्ट्रैक्टाइल एलिमेंट ऑफ अ मसल्स. जबकि इंटरमीडिएट फिलामेंट में हम देखेंगे ये एक्सटेंड अक्रॉस साइटोप्लाज्म कनेक्टिंग डेस्मोजम्स एंड हेमीडेस्मोजम्स, द न्यूक्लियर लामाइना इन स्किन एपिथेलियम आज केराटिन. सो लोकेशन इन द सेल कहिले इंटरमीडिएट फिलामेंट कोठे सब अच्छी, द न्यूक्लियर लामाइना इन स्किन एपिथेलियम आज केराटिन. ठीक है. अच्छा, माइक्रोट्यूबुल भी एक है. ठीक है. तो माइक्रोट्यूबुल का शेप क्या होता है? लॉन्ग होता है, नॉन ब्रांचिंग होता है. डायमीटर कितना होता है? 25 नैनोमीटर. बेसिक प्रोटीन यूनिट इसमें ट्यूबुलिन पाया जाता है. लोकेशन इन द सेल कहाँ पर माइक्रोट्यूबुल प्रेजेंट होता है? माइटोटिक स्पिंडल में एंड कोर ऑफ सिलिया में प्रेजेंट होता है. मेजर फंक्शन माइक्रोट्यूबुल का क्या होता है? प्रोवाइड्स नेटवर्क फॉर मूवमेंट ऑफ ऑर्गेनेल एंड मूवमेंट ऑफ सिलिया. नाउ filamentous actin help in movement of a chromosome and cell division. Though anchoring protein F actin fiber attached to various cytoskeletal structure and interact with membrane bound protein, microfilament project into the microvilli present on the epithelial surface such as on the intestinal mucosa and extended up to the tip of microvilli. They help in microvilli movement. Microfilament in the lamellipodia मतलब laminar extension from cell that occurs at the time of a cell crawling helps in cell movement on a surface. Actin filament interact with integrin receptor to form focal adhesion complex. Focal adhesion complex serve as a point of traction with the surface over which cell pulls itself. Microfilament are also used by some molecular motor as tracks. Then microtubule. Microtubules are long hollow tubular structure figure 4.11c. कहाँ पर? Okay. Microtubule. Microtubules are long hollow tubular structure having diameter of about 25 nanometer including the wall thickness. The inner cavity diameter of a microtubule is about 15 nanometer. They are made up of subunit of a globular protein called as tubulin that are arranged in a closely packed helical manner. Tubulin is in microtubules are of two types, alpha tubulin and beta tubulin. Except the tubulin in centriole that are gamma tubulin. The alpha and beta tubulin subunit from the heterodimer that aggregates to make tubular structure, that is protofilaments of a stacked ring. Each ring in microtubule usually contain 13 subunits, that is 13 protofilaments. Special characteristics. A unique property of a tubulin subunit is their property of disaggregating मतलब disassembly and reaggregating मतलब assembly. Therefore, microtubules forms a dynamic cytoskeletal framework of the cell. Microtubules are polar in nature with assembly predominating end मतलब positive end and de-assembly predominating end, that is negative end. The process of assembly and de-assembly may occur simultaneously. Many drugs act by inhibiting microtubule assembly. Clinical works 4.4. Interaction with GTP facilitates microtubule formation. Microtubule growth is temperature sensitive with cold condition favoring dis, disassembly. In cell with cilia and flagella, microtubules extend into these structure. Kinesin and dynein are microtubule based molecular motors. Clinical box 4.4. Cytotoxic drug act by inhibiting microtubular function. Many cytotoxic drugs act by inhibiting the microtubular activity. For example, anti-cancer chemotherapeutic agent such as vincristine, vinblastine promote disassembly of microtubules. Paclitaxel, another anti-cancer drug, bind with microtubules and stabilize them against depolymerization. The stabilizing microtubules function abnormally and do not undergo dynamic change necessary for cell cycle completion. It prevents formation of a mitotic spindle and cell die. Colchicine inhibit microtubule assembly. Structure of A, that is microfilament. Structure B, intermediate filament. स्ट्रक्चर सी आपका माइक्रोट्यूबुल. स्कीमेटिक रिप्रेजेंटेशन ऑफ अ डिपिक्टिंग द फॉर्मेशन ऑफ अ माइक्रोट्यूबुल. अल्फा सब यूनिट ये हो जाएगा बीटा सब यूनिट. 13 प्रोटोफिलामेंट फॉर्मिंग द वॉल ऑफ ए माइक्रोट्यूबुल. उसके बाद फंक्शन स्पेशल कैरेक्टर्स माइक्रोट्यूबुल. फंक्शन. Microtubules serve as a cytosolic guide rail for transport of substance within the cytoplasm or transport of secretory granules to the cell membrane. They help in transport of vesicles and organelles from one part of the cell to another. They contribute to the maintenance of cell strength and cell shape. They help in the formation of spindles that move chromosome during mitotic cell division. They are pillars of structure and function of cilia. They contribute to cell motility. Cilia. Cilia are hair-like outgrowth of cell of the epithelial membrane that characteristically beat in synchronous whip-like fashion. Therefore, cilia allow a directional flow. Each cilium is an outgrowth of a basal body situated beneath the cell membrane, which has the property of reproducing itself. In each cilium, there is an axoneme that typically consists of nine doubtlets of microtubules that run circumferentially from base to tip, while a single pair of microtubule runs centrally, that is 9 plus 2 arrangement. All microtubules in cilia are linked together by protein cross-linkage that forms the axoneme. In cilia, dyneins are present close to the axoneme called as axonemal dyneins. The coordinated interaction between the tubulin of a axoneme and axonemal dynein promotes ciliary movement. Basal body is present at the base of axoneme, which has arrangement of microtubules in nine set of triplets similar to that of arrangement in centriole. Then ciliary movement. A whip-like movement of cilia called as a ciliary movement that occurs on the surface of the cell is important at two places in the human body. Number one, the respiratory tract. Number two, fallopian tubule. The cilium has the appearance of a sharp pointed straight or curved hair that projects 2 to 4 micrometer from the cell surface. Cilia are usually many in number. For example, there may be as many as 200 cilia on the surface of each epithelial cell in the respiratory passage. The cilia beat faster. The cilia moves forward with a sudden, rapid whip-like stroke at the rate of 10 से लेकर के 20 times per second, and then it moves backward slowly to its initial position. The rapid forward movement pushes the fluid or content in the lumen in the forward direction. The slow dragging movement of cilium in the backward direction has almost no effect on fluid movement. As a result, fluid is continuously propelled in the direction of the fast forward stroke. Most ciliated cells have large number of cilia. They are oriented in the same direction. Therefore, ciliary movement creates an effective means for moving content in one direction. The ciliary clearance of the content in the lumen, such as mucus, is impaired in the ciliary dyskinesia. Clinical box 4.5. ओके ये है स्टेरियोसिलिया and cilium in hair cell of a vestibular apparatus are other example of cilia on the surface of the cell in the body. The flagellum of a sperm is similar to cilium as it has similar structure and same type of a contractile mechanism. However, the sperm flagellum is much longer and moves in quasi-sinusoidal waves instead of whip-like movement. Clinical box 4.5 Ciliary dyskinesia. Ciliary defects are usually of a primary inherited disorder affecting the ciliary movement and are grouped as primary ciliary dyskinesia. Respiratory system is usually affected. Altered ciliary function in the conducting airways decreases the mucociliary escalator and result in airway obstruction invariable associated with infection. Dyskinesia of cilia, flagellum of sperm result in infertility. तो आज हम यहाँ पर इसको एंड करते हैं. आज का डेट कितना है? 16/1/26. Thanks for